So hello there, this is Anurag Pradhan, and in this video we'll be studying about PCR. PCR. So the full form of PCR is polymerase chain reaction. Polymerase chain reaction. So now the question arises, why we use this technique? So let's say we have a DNA. So the piece of DNA is called gene, and then we want to make multiple copies of this gene for some reasons. So the classical method used to be just take a vector and then put this gene on this vector and then use a host organism. Let's say, for example, we take this bacteria, and then we are putting this vector inside the bacteria. So what bacteria will be doing is just making multiple copies of this vector, and then we can extract these multiple copy of vectors, and then we can extract our gene from these vectors, thus making multiple copies of this gene. Okay, so it was like in vivo condition, and then like what if we want to do in vitro conditions? So there is a scientist, there was a scientist named Gary Mullis. He came up with a very good idea, the idea which revolutionized everything. For this idea he got Nobel Prize. Okay, so like PCR, this technique, the idea for this technique fetched Gary Mullis a Nobel Prize. So like it was really a big discovery in the field of biotechnology or biology or biological sciences, whatever you say. Okay, so in order to understand how this technique works, initially we need to know what are the components or the material required for using this technique. Okay, so components or material required for using this technique. Okay, so it's obvious, like, you know, if we want to make multiple copies of this DNA, we must have some nucleotides. So for obvious reasons we require nucleotides. Then we require a buffer solution. And it should be nucleus free, you know, because what if there are some chemicals inside the solution which can degrade DNA? So like it should be nucleus free. Nucleus free means it should not have any type of chemicals which can degrade the nucleotides. And then we require primer, RNA primer, and DNA polymerase. So these two were like obvious choices. So why we require RNA primer and DNA polymerases? And you must be wondering which type of this RNA primer is. So actually, let's say we have a DNA which we want to amplify, and it has a certain sequence, let's say A, G, T, C. It starts with this sequence. So in order to design a primer, you know, like you must be wondering like why we are designing the primer. I'll be telling very soon. Okay, so let me just tell you that how this RNA primer is designed. So RNA primer is designed in such a way that it has complementary bases. For example, let's say this is from our DNA part, so the complementary bases will be okay. So our RNA primer will be having this sequence so that it can attach at the starting point or the beginning of this DNA. So why we are talking about this? Okay, why we are talking about this? So how this technique works, let's get its insight. We have this rDNA, and then the first step for using this technique is raising the temperature of this DNA solution to 96 degrees Celsius, approximately 96 degrees Celsius. Okay, why we are raising the temperature to 96 degrees Celsius? So we are raising the temperature for denaturation of DNA. Denaturation means making this double-stranded DNA or converting it into single-stranded DNA. So why we are converting it to single-stranded DNA? So the answer to this question is in step 2 and step 3. So step 2. In step 2, we lower down the temperature to 55 or to 65 degrees Celsius. So why we are lowering down the temperature? So in first step, we just separated or made single. In the first step, we converted the double-stranded DNA into single-stranded DNAs. What we want to do is just attach the primers on this split or single-stranded DNA. So let's say our primer is getting attached here. So our primer can attach at low temperatures to the single-stranded DNAs. So now our RNA primer has been attached. Now we want it to attach DNA polymerase, and we want it to synthesize the further part of this single-stranded DNA. So now at step 3, temperature is again raised to 72 degrees Celsius approximately. At this temperature, the DNA polymerase will bind to RNA primer, and then it will be extending this part. So here we had this RNA primer, and now we have DNA polymerase which has been binded to this part, and this will be extending this single-stranded part further. So this, in order to extend this part, we also require nucleotide. As I said, nucleotides are required obviously in order to extend this part. So I think it makes sense why we require nucleotide, why we require buffer solution, and why we require RNA primer and also DNA polymerase. And one important thing, please, we also require template. Template means the DNA sample or the gene which we want to amplify. So these are the requirements for the PCR chain, polymerase chain reaction. So that's it. And one more important thing. So you must be wondering, as here you can see that the temperature is 72 degrees Celsius. So how this DNA polymerase is working at such a high temperature? So the answer to this question is that we'll be using a DNA polymerase which can sustain such high temperatures. So we get that DNA polymerase from some bacteria which can sustain such high temperatures, and most commonly we use Taq polymerase. It can really sustain such high temperatures, therefore we use this polymerase, and this is how the gene can be amplified. So let me summarize everything. There are 3 steps: step 1, step 2 and step 3. In step 1, the temperature is raised to 96 degrees Celsius. In step 2, it is then again dropped to 55 to 65 degrees Celsius, and then it's then again raised to 72 degrees Celsius. So this first step is denaturation. Denaturation is nothing but converting double-stranded DNA into single-stranded DNA. The second stage is called annealing. Annealing means the binding of this RNA primer to this single-stranded DNA, and then the last step, and this process is called synthesis, because here we are synthesizing the single strand, another single strand, in order to make the final double-stranded DNA. And then again this cycle is repeated to get multiple copies. So this way our DNA will be multiplied several times, and the production will be in exponential form. So that's all about PCR. Thank you very much for watching this video.